Welcome to the Terminology Leading Practices stream of SNOMED CT Expo 2020. I'm Fleur McBriar from SNOMED International's Communications team, and I'll be moderating today's session. Please use the Q&A box to type your questions to the presenter. All questions will be answered at the conclusion of the presentation. I'm now pleased to introduce Sarah Harry, a Senior Clinical Terminologist at Templatebased Limited in the UK. Sarah will be presenting a session about bridging the gap between local lab code test lists and central reference standards. Sarah, please proceed.
Hello, everybody. My name's Sarah Harry, and I work as a clinical terminologist for Templatebased Limited. As said, we're a small startup company specializing in user-friendly web applications in healthcare, led by Jay Kohler. So standardized, user-friendly laboratory medicine test results, bridging the gap between local code lists and central reference standards by interfacing through intermediate representations. My history is as a hemodialysis nurse, followed by UK commissioning performance management roles, before finding my ideal job as a clinical terminologist. I managed the UK NRC authoring team for a good while and oversaw the PBCL lab results list in the UK as well. And I'm going to briefly run through our work on analysing and standardising these intermediate formalisms for lab results reporting using a web application and templating. Okay, so why would we attempt this tool? Because there's a gap. There's a gap here. There's a gap between locally curated lists of results and the formalisms of SNOMED. SNOMED has the scope to be an interchange with any lab terminology through its comprehensive test procedure, observable entity, result structure, leading through to test findings and diagnoses. But we have this gap, and what we see here on the left is the local descriptions that you would see in a local laboratory, where they've just got a simple list of test codes that will come from various procedures that they run, and it'll be something simple like this, AST. It may have a code, maybe the same thing there may be a numeric code, but they work on these simple lists. There's other information in there as well. This isn't all they use, but it's just to show that the relative basic level at which this operates. There may be a unit of measurement column there. There'll be some other columns as well, but this is the body of it. On the right-hand side, we see the SNOMED CT FSN that might be an equivalent to this, and that's catalytic activity of aspartate aminotransferase in serum. Now, there's clearly quite a substantial gap there in terming, and that's really the gap that we think needs addressing. And I've also put in a few extra examples of these down on the left for bilirubin and C3, C4 and things like that, which are all of a similar format. And this is just picked out at random, and this is the sort of thing you'll find. So what do we do here? Well, we could just create lots of SNOMED CT-based concepts for all this and then just map it all, yeah, with translation maps. Mapping can be a pretty horrible exercise. It's a very attractive idea, but in practice it can be pretty horrible. The idea of one-to-one maps is a great idea. In practice, it isn't always that simple. We've got a creation and maintenance issue. It's not necessarily one-to-one. It could be one-to-many. There's a possibility of semantic loss. There's a great deal of overhead involved in maintaining these. So it's not a straightforward idea. We think there are better ways of doing this. So the excellent formalism then that SNOMED employs with a full set of attributes and comprehensive value sets can be cumbersome in direct use. So the terms are long, formalistic, and they need to be, that's fine. But those who report results generally want to keep to local lists, to simple lists, even reduce to simple lists of substances and units of measurement. And they can do this because within their domain, the tacit knowledge that they have, that they interpret these things with is huge, and it underpins these terse forms, so that's okay. So we have this tension between the lab senders and the clinical receivers of those lab tests wanting to retain their lists, and the commissioners for lab results messaging require a standardization of SNOMED. So how do we deal with that? Well, the solution from our perspective is to allow experts to create standardized terms, things that expert terminologists understand, that lab users can understand, without having to, having to understand the whole of SNOMED CT formalisms. And one of the aspects of that, besides the intermediate forms here, is smart templates. We need some sort of interface in which we can manage those intermediate forms. So smart templates are the thing here. Well, what did we do? We created a model then for embedding domain knowledge in our templates and in these intermediate representations. So we've got easier authoring and review using smart template-based content creation, producing terms and concepts that are more user-friendly, allowing better retrieval of appropriate content. And diving straight in to keep you waiting on this, let's have a look at just what that actually means, and then we can explore it a little bit further afterwards. So here we see a screen grab from our PathNexus tool, from our online web tool. And this is an editing environment that any clinical author would see. So it's an edit concept window. And from that, in order to add a concept, you would want to pick a template to use. And this is simply a drop down of templates. There's some familiar ones there, which would be property of component in system using technique, but some extras there, some more specific ones: antibody, antigen, enzyme, substance concentration. So we can pick one of those, which will help to constrain our values. What does that mean then? So simple templating. We'll look at the smart templating in a bit. Simple templating. So you see from that edit concept window, we've selected a template here. We can select a component, and here it's the U1 ribonucleoprotein, specimen, serum, and just add that in, and the property. And here it's actually constraining it for us. So because we've picked this antibody presence observable, we've got it's constraining it to just a present property. But there may well be a choice of quite a few here, which would be rather tricky to kind of figure out which one you're supposed to use, and it's prone to user error there. But we'll take the presence one that's offered. That adds in the presence property here. When we're happy with this triad that we've got here, we can generate. We can click generate here, and what this does is generate the fully specified name, the preferred term, and the PathNexus terms for this concept. So we've got the standard FSN up here as it is. So presence of, and the antibody there, same with the preferred term. But we also have this PathNexus term where we've abbreviated the antibody down to U1, SNRP, and AB. We could mess with these things further. We could abbreviate presence, we could abbreviate serum. But essentially, this is our intermediate form. There's nothing mysterious here. You can see these in other terminologies as well. But what we have done here is we've associated this short form with the SNOMED concept. And we can then relate these back to the forms that people are using. So we can save it, and that would take it to our repository in a coded form. So what's going on there? So smart templates use these intermediate representations. Well, those simple templates do as well. Imagine this as a weaker formalism, if you like, a domain-specific language that allows users to create concepts tied to the SNOMED formalism behind the scenes. And there's some experience from the Galen project, which preceded a lot of the SNOMED work on this, around that approach. And it uses underlying domain knowledge that often users are trivially able to provide, such as units of measurement. So they can provide the domain knowledge that allows us to inform this content. And the advantages, well, it's easier for authoring. It will eliminate errors. It standardizes forms. It produces terms and concepts that are more user-friendly and allows better retrieval of appropriate content. So what did we do? Well, we looked at how SNOMED interacts with all those other terminologies. The UTL, the Unified Test List in the UK, LOINC, MPU, the nomenclature for properties and units. And we reviewed the attributes used and identified the read-across commonalities between them. We were then able to identify a core set of these attributes as the basis for an intermediate form. Just worth remembering that there will always be some philosophical differences between terminologies. There's not always an exact match between these things. They won't overlay precisely, but we're looking at what the commonalities are. What's the reducible commonalities we can use? Now, when we looked at the UK work on UTL development here as a successor to that PBCL list that I used to curate and that's overseen by NHS Digital in the UK, we see that the numerous available attributes mapped onto very similar or exact matches across terminologies. And that's no surprise, really. A lot of people who work in this area will already know that. So it confirmed some of our thinking on this. Now, the lab terminologies generally align or even specifically derive from things like UPAC, metrologic, ontologic principles. Which I don't have space to go through here. I will simply note there that the C in UPAC, of course, is chemistry, and not everything is chemistry. So there are still some other things there, some little issues to iron out here and there, but it's one to park at the moment. In general, it's a very robust model. But what's clear is that not all attributes are equal, basically. And apologies to those who are very familiar with this work, who've worked in this domain, will be only too familiar with this, but I'll just quickly show you the attribute set for observable entities, because not everybody will have seen these. So there's 20 of these in all. And you go, what, 20 attributes for observable entities? But bear in mind, though, that observable entities are split 4 ways between quality, disposition, function, process. So we can already start to think about maybe some of these we don't need to include. We're in the realm here in the lab field for these specific sort of blood science types of quality, because quantity is a type of quality. No, those aren't a binary. Quantity is a quality ontologically. So does the average clinician understand some of these? I don't think they would, and sometimes terminologists don't even understand some of these. So in, here's in, has realization. These are things that actually mean things in a very rarefied way to specialists. But what we've got to do is actually help people to work their way through this without having to wrestle with these things. And the work is very much derived from the UK work on PBCL, UTL, MPU, and LOINC. So what can we expose? What can we hide? We can already knock out quite a few. We can take out significant ones here, which I've grayed out, which leaves us with a list of things like component, in, here's in, property, scale type, technique, time aspect. And I've also picked out a couple of others there that are of interest, which I'll note and then put aside, which are precondition and relative to. So we'll just, those are the things that we'll see. So if we reorganize those, what we actually get then, and some people will be only too familiar with this, as I am, that this core set of property component and inheres in, and that cognitive triad most users will understand. And this for example, would be something like, if it was sodium, you would have something like a molar concentration property of sodium component inheres in serum specimen or something like that. So that's an example. And so that's pretty basic and common across all terminologies. Yeah. What we then have, though, when we look at those terminologies, we also have this secondary set. And you can see there's this set of 6 here, and people who know LOINC and MPU will see the commonalities across these things. Scale type, technique, and time aspect. Now I'll just have a bit of a more of an in-depth look at those, really. Scale type. So I'm not sure with some of these, really, because I'm interested in whether things are definitive or derivative, and some of these things look more derivative or look like metadata, and or things that have a specific purpose but aren't necessarily part of SNOMED concepts, which have a fairly constrained set of attributes that we need to model them with. Can it be inferred from something else? Well, yeah, you can actually tell what scale type is from the property. So if it's mass concentration, you would know what the scale type is. And in fact, from the user end, if you know that it's milligrams per liter as a unit, you know what the property for that is as a mass concentration, and that will take you there. So we can populate it from that end. I'm less clear about how that is some kind of definitive thing. But we'll put it to one side. I'm sure there are people who would be able to tell me all about why this is vitally important. And scale type, just by the way, is that Stevens set of ordinal, nominal, ratio, and interval that has been around for a long time. Technique and method. Clearly very important, very useful, indeed sometimes essential for test interpretation at the moment, very important around the dreaded COVID-19. So important stuff, but it's not essential. I think that's the point here. And there is, don't forget, there is also an HL7 version, whatever, FIRE environment for these to find space as well. Not everything has to go in the SNOMED concepts. We can find other ways of representing these, but putting them all together into an interface where somebody wouldn't see any join between them, if you like. Okay. So it can cause quite a lot of content bloat and noise, really, if you start sticking method on the end of everything like sodium that really doesn't need it most of the time. So we've got to be a bit careful with this. Time aspect, an interesting one as well. I thought this might help in solving that elephant in the room problem of test preconditions of things like fasting glucose and 30-minute glucose. But it's actually used specifically to describe mainly point-in-time test in the blood sciences field anyway. And those that aren't point-in-time tests are 24-hour results. Now, this means something like if you might have 10,000 codes, about 9,800 of those would be point-in-time tests, and then you've got some 24-hour ones. So again, it's another one I'm quite skeptical about why we have to use it as some defining thing. By all means, we can keep it in, we can populate it, but I'm not sure why it needs to be there in a SNOMED context. And there's various ways in which time dimensions can be modeled and messaged, and we can look at ways of doing that. Anyway, all the rest, I mentioned units. There are columns like domain specialism, so hematology, chemistry, microbiology, that sort of thing. Clearly that's metadata, useful for cataloging, but it's not part of the SNOMED concept. Additional term forms, as I said, the abbreviation that I showed you, there's nothing special there. LOINC and MPU have those things as well. There's long forms, short forms, date of activation, date of inactivation, all the rest. So that's not part of what we need here. What I did draw out, though, was those two things, relative to and precondition. Relative to is worth mentioning because I don't think in general our community has addressed this denominator issue for substances and cells in particular in ratios and percent counts, and I think that's just something worth marking for discussion. The other one is the precondition. Now there is actually an attribute for precondition. I'm going to park that as well. There are a lot of content that does actually have this sort of precondition in it, marked with a caret symbol or things like that. The main ones that are in there that need to be addressed are those timed tests, but this precondition thing does spill over into indications and metadata and orders and all sorts of things, and I think it's unconstrained and it could just be a catch-all, so I'm reluctant to go there really into picking that one up. Anyway, bit of in-depth analysis there. Translating all that into the user experience. Okay, so in summary, we've got these lists of components, properties, specimens, methods. We can now create the combinations of these that look both ways, down to local list forms and up to those national, international standards. We can abbreviate the forms so that they're familiar to people, and yet these forms still have their SNOMED equivalents. So instead of trying to cross-map between all these shifting ontologies, we can create a SNOMED model-compliant display that remains user-friendly. We'll just take a bit of a look at the templating world. SNOMED International have a templating service available. They've been working for a number of years now in the authoring service and the authoring tooling to allow for template-based authoring, and it's a really great piece of work. The templates are available online examples, and they're open source, so there's plenty to go out there, and I'd recommend that you have a look at those. It does need, for this specific field, to localize things. We need some terminology and some lab medicine understanding to enhance these, I think. Now, enhance them. Why enhance them? To avoid having to fill out every slot in a template, as I showed you before. For a lot of users, they don't know what property it's supposed to be. They may be confused over these things around terming. What we can do is actually take some of the heat out of that, and we can actually even support lab users and clinicians to actually create content if it's constrained. So it's not just for specialist authors, if you like. We can extend the engine to create user-friendly terms, embed domain knowledge, and there's also on-the-fly retrieval of concepts, but that's not really part of this presentation. So what we've done then, we've used SNOMED International's open source template syntax and its expression constraint language, and we've built these observable entity templates that I showed you earlier for the lab domain and the observable entities. The current template design that SNOMED International have got is great, but it's rather naive, and naive in the sense of simple. They're perfectly good. There's largely a set of passive field constraints, and I think that's fine, but I think we can go further, and we have done. We've done some further work on waking these templates up, if you like, and building smart templates that can infer, suggest, support associations for content with for users driven by intermediate forms then. So this is going to also include machine learning so that you can actually help this interaction improve over time. And I'll just show you then what does that mean then really in terms of smart templating. So this is a slight advance on what you saw earlier, and here, instead of actually selecting a template, we're simply asking users to identify the component and how they report it. That's all we need from them, because we can actually get the rest from domain knowledge. So if somebody selects the component, and here it's the same one that we had before, so we've selected that. If we select the specimen, it could be a default specimen, doesn't have to be serum there for some of these things. And then we just need to say, how do you report this? And it could be reported as a titer, antibody index, international units. But Somebody says, no, we report that as a Boolean. That's a positive, negative, or some form of neither positive nor negative. So we'll select Boolean. And notice here what that's done now is it's loaded up the template. It said, I know what this is. Here's the template. I'm going to populate the property. And I'm going to make all this look right over here in the terms. So when we click generate, it's going to create them in the right way, but it'll look the same as you've seen already in the other templates. So we've got the FSM preferred term and a path nexus term, which is our intermediate form, and click save, and that's saved as a SNOMED coded concept. And we've actually supported people through it. So what we've also got briefly is in our web application is looking the other way. We've got a local lookup service, so that can set one-to-one or one-to-many associations with those intermediate forms. And here's a window for this side of it for retrieval. We can either make these associations to existing published content or create new content. Users can load local lists. Content developers can load lists of substances or cells, etc. that need adding. And these SNOMED concepts and expressions then can just as easily align with any other lab terminology, whether it's SNOMED aligned or not, because the basic attribute value sets are in alignment and where it matters. Yeah. So here, somebody's loaded up a local list of their lab things. So it will just be a spreadsheet. It will get loaded. One of those in their list is sodium. So they click on sodium, they say, please find possible associations. And here it's popped up with some familiar ones that you'll see of sodium concentrations in a range of fluids, one or two that shouldn't be in there. So something like sodium valproate. So we don't want that one in. So we can, on the right, then assign whether they're exact matches or less or more specific or no match. And we can save those, store them with the local lab lists so people can actually manage their lists internally and update them as they wish. Okay, so I think that's just about it. We think those intermediate forms are something that are near enough to local lab forms to be usable directly, but if people just want to use these to create associations in the treasured local lists, that's fine too. Either way, we're supporting SNOMED implementation by helping those who don't need to or don't want to get involved with the complexities of SNOMED, and we're doing the heavy lifting so they don't have to. My name is Sarah Harry, and I'm a terminologist. Thank you for your time, and over to questions.
Thank you, Sarah. Can I ask that attendees give us a moment while we switch over from our pre-recorded content, and then Sarah can answer your questions. Please also note that after the live broadcast, this session will be available on the event platform for the next 3 months. Thank you.
Hi, welcome back to the live session. First, I would like to thank Sarah for the presentation, and there's quite a large number of questions, and we try to cover as much as possible. First, it's about when you're doing the simple template for the U1 SRNPAB, does the user presume specimen antibodies could also be reported in titers? How would the template differ for antibodies?
Yeah, interesting one. I don't know if I think the later smart template thing might have shown the solution there, because what you're doing is you can, from what the user inputs, there will be a presentation of possibilities for what is represented. Whether that's serum, or you could just as easily say, well, it's the patient as system. And there's obviously a clinical choice there to be made about what you really want to state. But by default, it would be serum. But the template would help you through that decision process.
Okay, thanks. I think some people are interested. Is that possible this template approach can be applied to other domains, for example, radiology imaging?
Oh, for sure. It's reusable. So you can take any SNOMED coding here. If you wanted to represent something like, CT of abdomen and pick whether it's with contrast or not, if you've got those expressions locally and you want to pick up the SNOMED codes for them, then it's very easy to just reuse this structure for that sort of thing. No worries.
And the template, I think here questions about related to SNOMED CT observable, and this reuse the LOINC part, SNOMED CT aligned with LOINC terms, and for the post-coordinated expression. Or this is something completely different, and any pro and cons versus LOINC.
Yeah, the analysis is based on LOINC and on MPU and on the UTL and PBCL codes in the UK. So all of this refers to those products and is structured in a way that it can be easily related to those. It doesn't relate to those expressions. I think here we're reliant on pre-coordinated SNOMED. But using the observable entity model, and so those 2 times 3 things can be mapped across all those terminologies. The expressions I think are helpful here, but I think that's where the difficulty comes, that we actually do need some pre-coordinated content in this area, whether that's in extensions or in core, and if that helps. Thank you.
Some ask about the SNOMED CT released, the beta release expressions for the LOINC attribute. In your design, have you utilized those for the template, or at least help the template the work you're doing?
Yeah. Well, we've analysed those. We've taken those things that are LOINC parts, that are MPU, that are in use in the UK, and come down to lists of those things that we can actually then map on. So this is actually a means of the user being able to have SNOMED representations that are common to all those standards and products, so that you can then map those SNOMED identifiers on further to things like LOINC and MPU. So sort of yes and no.
Thank you. The last question, we overrun a little bit. Could this be developed to also use for correction of the errors, or at least improve the quality in the current content?
I think that's really interesting, because what this does is bring the SNOMED forms closer to the user, so they're much more able to see when they're making associations between the local terms and SNOMED. If you search for something and it returns content that you consider outdated or bad or just not there, then there is then a means of actually going to SNOMED International or your NRC and saying, hey, this needs changing, we need another code in here. So yeah, it would be an excellent thing, an excellent interface for that to happen, I think.
Yeah, and I also added a little bit in SNOMED International. We also utilize template actually as part of QA. I think, as Sarah mentioned here, the work possible you can expand for different use case. Yeah.
Yes. Certainly could be, and for updating things, because you can do all sorts of bulk updates and bulk checks on things. So yes.
Okay, I think that's all. Thank you all for the participating in the QA session, and enjoy your time at the expo. Thank you. Bye.